Today I'm going to talk about who we are and who we will become, and how the field of developmental cognitive neuroscience will start to answer these questions. And I usually like to start with a video of a newborn, right? So as you can see, this newborn is sleeping. Newborns sleep a whole lot, right? And you can see that her eyes are moving around. She's probably experiencing REM sleep, maybe dreaming. The question is, what is she dreaming about, right? She was just born. What is she thinking? What does she know? And who is this person going to become? And these are questions that humans have asked themselves for centuries, right? What makes me me? And is it what I was born with, or is it the experiences that really shape the person that I am today? So it's the classic nature versus nurture debate. So to formalize these questions, right, how much of the human mind is determined at birth, and how do we gain new skills that are uniquely human? Is there a limit to our potential based on the stuff in our brain that we're born with? But what if I were to tell you that science is now at the point where we can start to really answer some of these questions? We're able to do experiments that we could only dream about doing 40 to 50 years ago. So let's see if we can answer some of these questions with brain imaging. And so here's an MRI scanner. I don't know how many of you have been in one of these or had the opportunity to see one, but this is what it looks like. You climb in here and you go into the scan bore, and we can get these types of images from it, right? So here's a baby scan. You can see brain anatomy, like where stuff is in the brain. You can get brain function images, so what each brain region is doing when you're performing a mental task inside of the scanner. And you can get these really cool connectivity images of what brain regions are connected to one another, the strength that they're connected to each other, and you can visualize all of these pathways in 3 dimensions, which is amazing. So using all of these neuroimaging methods, here's what we know about the adult brain. Right, so here is an adult's brain, and I inflated all of the crevices such that we're looking at the entire surface of the brain unfolded. And we turn it around such that we're looking at the underbelly. So the eyes should be here, the back of the head is here, and we're looking at it from this way. So we're looking right here, in the temporal cortex. And we know that the adult brain has a very rich sort of organization, and there are specific brain areas that do very specific mental tasks. So this is one of my favorite brain areas. These brain regions help us perceive faces. So whenever you're looking at a face, whenever you're looking at my face, right, these are the brain areas that are engaged and activated. And if you lose these brain areas, like with stroke or any damage, you're unable to perceive faces, even familiar ones. And that's a condition known as prosopagnosia. Other visual areas are responsible for how we perceive everyday objects like chairs, clocks, whatever, right? All the stuff around us. Other brain areas help us perceive scenes. So when we're navigating through the world, when we come into a new place, these brain regions are active. It's like a GPS system that's built right into our brain. And here's a brain region that I'll talk about in some depth today. It's called the VWFA, or the Visual Word Form Area. And this brain area is responsible for helping us read. So it processes orthography. Other brain areas help us understand what it is that we're reading. So these are the regions in yellow, and these are the language areas, right? So they help us know what people are saying, they let us talk, and they let us communicate with one another. Apart from these rather domain-specific brain areas, we also have brain regions that are more general. And these regions help us attend to things in the environment, and they help us filter out irrelevant things. So these regions are more general purpose. Whenever you're focusing hard on something, when you're trying really, really hard, no matter what it is, right? It could be math, it could be trying to read something, understand something, listen to someone. These regions are activated if you're focusing hard. All right, so does this organization exist at birth, right? So all of these brain areas are present in every person that we scan, and they're in approximately the same place. So, it's kind of weird, especially that some of these brain areas show up in the same place. So we're wondering what drives this functional organization of the brain. And does this organization already exist at birth? Are we born with it? And we're now at the point where we can peek inside a baby's brain and we can compare this brain to other brains, and we can use computational modeling and artificial intelligence to try to predict things like future outcome and behavior in each individual baby. So imagine being able to know what your child is thinking about and how you might be able to help them reach their highest potential. It would truly be revolutionary. And so that's what we're working on, but it takes a lot to scan kids, right? It's hard and not every lab can do it, but the questions are just too interesting to not try it. In my lab, when I got here, we spent quite a bit of time trying to figure out the best way to collect high quality data, and we've converged on a set of protocols that we now implement. So for newborns and infants, we wait for them to fall asleep and then we scan them. We swaddle them, mom feeds them, and we wait for them to fall asleep. And once they're asleep, we move them to the scanner and an experiment. This is me here. An experiment is with the baby throughout the whole scan. For children, for a bit older children, we do as much as we can to prep them before they come into the MRI. We send kids a video to watch before they come in, and we've modified our video slightly for the current pandemic state. So here it is.
Welcome to Z-Lab, the Sagan Developmental Cognitive Neuroscience Lab. In our lab, we study how kids like you learn and remember things, how plasticity happens in the brain as we learn new things, how it makes us into different people, and how it is able to recover from any injury that might happen to it. We learn about these things when kids like you participate in studies like you're doing today. We're going to show you what to expect with an MRI scan in our lab. So first, after picking you up in the lobby, we'll take you upstairs to our lab. And this is our lab here. This is where we'll learn the games and fill out some papers. While in the lab, during the pandemic at least, we'll take your temperature like this. In the MRI scanner, you'll have to wear earplugs because it's really loud. We'll have you practice in the lab, just like Athena here. She's one of the researchers here at OSU, and she might even be the one helping you today. After we learn the games, we'll go from our lab to an elevator that will take us downstairs to the MRI scanner. But before we go in, we'll make sure you go to the bathroom one more time just to be sure. And then you'll meet Darren. He runs the MRI scanner and ensures your safety. To make sure you're safe to go into the scanner, he'll use a special wand to make sure you don't have any metal on you. You can see him checking our scan dog here, and upon ensuring there's no metal, he'll allow you to go into the scanner. You'll lay on the table like our scan dog here. We'll help you put your earplugs in so it's not so loud, and Darren will put a mask over your head so that we can get good pictures of your brain. And then we'll give you the buttons to push for the games. Then Darren will move you into the scanner like he does with the dog here. You won't be alone. Someone will be with you by your feet the whole time. And after the scan is over and we have all the pictures we need, Darren will move you out of the scanner one more time and you're done. You can go collect your toys and snacks because you helped us to do some science.
So yeah, kids get this before they come in, and after the scan, we give them compensation, but we also give them snacks and small toys, and they get them, and they know that they're getting them after they complete each short scan. So it's motivational for them. So in my lab, we try to answer the overarching question of how does the architecture of the mind arise? And we study children, neonates, and developmental disorders. And I'm going to give an example from each of these three branches of study in my lab, and I'll use reading as the overarching unifying theme. All right, so as we know, there's this region of the brain that's critically involved in reading, right? It's called the visual word form area, or the reading area. And what makes reading such a great place to start is that this region that's involved in reading doesn't exist before reading, right? It exists as in the brain cells exist, but the cells in this region don't know the difference between words and other visual stimuli. So we say that this region doesn't exist in pre-literate kids or adults. But there may already be the right type of connections that are set up in this region, right? Maybe this region is just waiting for the right type of input to become an orthographic reading area. So to answer this, we scanned kids before they knew how to read at age 5, and we got a connectivity scan on them. And then we brought the same kids back at age 7 after they learned how to read, and we got a functional MRI scan on them. And that was in order to delineate where the VWFA was in each child's brain. And we wanted to test whether early connectivity at age 5 could predict the exact location of the VWFA in that same child at age 7. So before I show you the results, we're going to inflate the brain again. And so remember that we're scanning kids before they knew how to read. And then we're scanning them again after they learned how to read. And we're going to see whether we can predict the location of the VWFA in each child. And the region varies in its exact spatial location in every child. So we wanted to know how precise could we be and how well we could predict its exact location in each child individually. So here's a child's actual fMRI data when they're looking at words and in the scanner, and word selective cortex should be here, circled in magenta. And we know this because we scanned them at age 7, and this is where word selective cortex is going to show up. And this is what was predicted using the same child's connectivity data alone at age 5. And what we can see immediately is that a lot of this word selective cluster can be very well predicted from the same child's brain scan at an earlier age. So we can use a brain scan at an early age to predict where in the brain of each child a reading area will show up. It turns out that this brain region is already connected to a set of other brain regions like language areas that set it up to become selective to written words. And so we're putting together a wiring diagram of the circuitry to better understand how it works and how it develops. So early connectivity seems to be earmarking the location of the VWFA, a region that's entirely experience dependent. Right? But this evidence wasn't entirely satisfactory. So next we explored whether this connectivity pattern was already present at birth. So when you're asleep, or even when you're just spacing out, as you may have done a few times already, your brain is still active and busy, right? So it turns out that regions that are involved in similar mental functions fluctuate together. So they're going up and down at the same rate. So what this means is that regions are connected to each other functionally. They're involved in similar mental functions. And it turns out that when a baby is sleeping, this is what's going on in that baby's brain. So this is a neonate brain scan. And so what we did was we compared functional connectivity in neonates to functional connectivity in adults. And we wanted to know whether this network, the circuitry, was innate. And we looked at the connectivity of these language regions. And what we found is that neonates, just like adults, have parts of visual cortex that are very well connected with language areas. And it so happens that that the language network connects more to what's later going to become this reading area, more so than all of these other areas. So it's not connected to face areas, it's not connected to scene areas or object areas. It's connected to what's going to become the VWFA. So a region that will later develop orthographic selectivity is already connected with language cortex even at birth. So pre-existing connectivity may be a way for a visual area to become language sensitive. Now I'm going to turn to the last example of today's talk, where I ask whether connectivity can predict later behavior and later clinical outcome. And specifically, we looked at whether we could predict which child would show reading difficulty or develop dyslexia. We know, for example, that there appears to be a circuitry inside the brain that's different in older kids who have dyslexia. So certain tracks, the ones that I've highlighted in color here, they tend to be different in older children and adults with dyslexia. One of the more important ones is this tract in yellow. It's the arcuate fasciculus, and it connects the frontal and the temporal language areas. So we wanted to know, are these differences the cause or the consequence of dyslexia? So are any of these tracks different in pre-literate kids who may later go on to develop dyslexia? If we do see evidence of this, it might mean that these differences are causal, and at the very least, they can be used as biomarkers of dyslexia. And what we found is that the volume of a particular white matter tract, the left arcuate, was correlated with a pre-reading measure of dyslexia, namely phonemic blending. So rendering a few subjects as arcuate fasciculi in 4 children, you can literally see this difference, right? So a child with lower phonemic blending ability has a smaller arcuate than a child with superior blending ability, and these are all normalized for head size. So the differences in this reading circuitry that we see in older children seems to exist even in pre-readers. And so we can make predictions about who might go on to develop dyslexia using a child's MRI data even before they know how to read. And this might help introduce early interventions and can potentially prevent issues with years of struggling to read. So I showed that early connectivity can predict the reading area location in each individual child. And that's pretty cool, right? Because what that means is that we can use non-invasive neuroimaging methods to predict what the brain will look like years from now. And that would be groundbreaking, right? And we also went even earlier and we found that neonates already have this rich circuitry set up, which suggests that innate mechanisms may determine what the brain might do years later. We then showed that not only can we predict the organization of the brain as it develops, but we can also predict learning and behavior using early brain scans. And this can potentially help issues with reading even before a child starts to have trouble. But we're just getting started, right? So we're continuing to do longitudinal studies on children starting as early as possible. And we're following up with them as they get older to see how well we can predict developmental milestones like walking and talking, as well as school age skills in each child using early brain data. And I talked about reading today, but we're also looking at other skills like math, attention and cognitive control, memory, emotional processing, and social processing. So we want to know, how well can we predict these milestones from early brain data? We also have some work that I didn't have a chance to talk about today that looks at how the brain changes across time, right? So how does experience and maturation change the brain, and what types of experiences lead to what types of changes in brain and behavior? But I'm going to take a step back and quickly talk about why this work is so important, right? So why should the world care about developmental cognitive neuroscience? So studying how the human brain and mind develops and making individual subject predictions has both scientific and practical importance. For basic science, if we can use cutting edge methods in cognitive neuroscience, and if we can combine it with cutting edge methods in artificial intelligence, and we can try to predict developmental milestones, we're going to establish where we are as a field in understanding how the brain gives rise to behavior, right? So we can't say that we truly understand how the brain works without being able to accurately make such predictions. But on the flip side, even if we are somewhat inaccurate at making these predictions, we will know where we are as a field and what we need to improve on, right? So we can actually test these things non-invasively in humans. This work provides a way to test the nature versus nurture debate, as well as understanding what about our brains make us unique, how we learn, and why childhood is so important, as well as what would happen to the developing brain if there were certain changes in experience. This research also has educational applications, right? Can you imagine if a brain scan could predict reading ability, math ability, and so on? So this could be a game changer for individualized and adaptive education. And the technology that's built from these findings could potentially shift existing educational paradigms. This work also has broad clinical applications, right? So if we're able to predict disorders even before they are apparent or even before the skill can even be measured, like in the case of predicting reading ability in pre-readers, we can try to develop interventions even before a child starts to have trouble. Interventions, remediation, and targeted practice will greatly improve the quality of life of individuals, and it might even prevent the development of certain disorders. All right, so this work is important. What is the future of this field? And I think that the future of this field is in merging big data with artificial intelligence, right? So using machine learning methods, much like the stuff that I presented here, to understand how the brain makes the mind and how it can change with experience and development. It's also in developing innovative technologies like MRI head coils and stimulation devices to make groundbreaking discoveries in cognition and development. The future is also an open and inclusive science. So we can't get to these goals alone, right? We need to collaborate and we need to share data, increase diversity, and greatly enhance scientific communication and education for our community. So what's the future of this field? It's OSU. So Charles talked a little bit about this, but I'm going to go from the faculty side and researchers side who's currently doing this type of work, right? So our department and OSU in general is a hotspot for all of this work. We have, as you can see, the state-of-the-art imaging facility right in our building. We have a high-performance supercomputing cluster at our fingertips. So it's the Ohio Supercomputing Cluster. And we're developing these cutting-edge technologies and making groundbreaking discoveries with award-winning faculty and researchers. So we have a really rare opportunity to be at the forefront of this new movement. And to help get us there, I'm leading an effort to develop a child study center that aims to put OSU at the bleeding edge of science and technology, and that will attract world-class talent. And I envision the center to be the catalyst for innovation and discovery about what makes us us and how we can use this information to make positive impacts on society. So in sum, cognitive neuroscience is now at the point where we need to be studying the developing human mind and brain longitudinally. And we need to work together to standardize procedures, pool our resources, and we need to take the time to dream up the best science possible such that we can inspire the new generation of scientists, policymakers, educators, and hopefully that we will be able to have practical advice for parents to help their little ones achieve their highest potential and therefore help shape the future of society. All right, so I would like to thank my lab. As you can see, we managed to get a Zoom background in nicely with our with our lab meetings. And I would like to thank my funding resources, and I'd like to thank you all for listening to me and to give me the opportunity to discuss my work with you.
Thank you so much. Oh, there we go. Thank you so much, Zainab. Really appreciate that. And as mentioned, this is an opportunity for an informal conversation about Dr. Sagan's work and for you to ask questions. So feel free to ask any questions you like, and Dr. Sagan can answer them.
So Zainab, first of all, welcome to Ohio State.
Thank you.
We're very glad to have you here. This is more of a clinical psychology question than a developmental psychology question. Obviously, with your training, your research, what have you, you probably could have gone to a lot of different programs, as well as maybe staying at MIT. What was in your thinking that made you select Ohio State?
That's a great question.
That's why I ask it.
Yeah, yeah. Well, I mean, some of the reasons that I presented, honestly, what makes OSU so great. So I knew a lot of colleagues who came from MIT to OSU Department of Psychology, and I kept in touch with them, and the department was growing and growing in terms of adding new junior faculty. And so that's mainly one of the reasons that I came here, right? So I wanted to have a good mix of both senior faculty that I could have as mentors and for guidance, but also junior faculty that I could interact with on a daily basis and who were very much into the, merging neuroimaging with computational science side of things. And the OSC, the supercomputer cluster, was another attractive one. It's pretty rare for there to be such a high performance cluster that it was free when I came. They're starting to charge a little bit now, but it's for doing this level of high processing, to be able to get it done in a reasonable time frame, to publish and to submit grants and such. We need, this work needs a cluster like that.
I have one other question. It's more operational than anything else. Obviously, working with children, younger children, the whole closed MRI is an uncomfortable set. It's uncomfortable for an adult, let alone a kid. Would this work be easier if you had an open MRI?
Yeah. That is a great question. I would need to look at the data quality of open MRIs. Right now, our scanner has, it's state of the art, and it has the ability to reach, high gradients, which is important for the types of connectivity scans that I do. So I would like to see that resolution. And if we can achieve it, that would be ideal. Anything that we can do to develop the technology to make it such that it's more convenient and easier to scan kids, I would welcome with open arms.
Thank you.
Thanks.
Hi, Zeynep. I have a question I'll go ahead and ask since no one else seems to be jumping in. As you are able to understand or potentially diagnose dyslexia, there are some treatments, I'm assuming, for dyslexia. And I don't know, do you plan in the future maybe to check their effectiveness maybe from a physiological standpoint and help validate which ones might be, I don't know, is this something that might be in the realm of your research in the future?
It's definitely a direction that I would like to head towards, yes. And as you mentioned, there are definitely treatments and practice that you could do. And it's unclear, right, whether there are different subtypes that might benefit from different courses of practice and intervention. And so it would, there is some evidence from other disorders that may inform this, right? So we can look at a let's say, cognitive behavioral therapy scan to say which individual would benefit from this type of treatment versus other types of treatment. And so I envision being able to do something similar with dyslexia as well as other developmental disorders. But for that, again, we need to follow up with them longitudinally, which we are, but it will take a few years to get to that point.
That's what drew me to wanting to hear you speak today. Is Dan and I are responsible for a 14-year-old grandson, and we are currently, I don't know what it is in Ohio, but we're distance learning, so schools aren't in session. So high school freshman.
Yes.
27 weeker, and it was at the end of 5th grade that the school finally said, oh, he has processing, his problem are processing. Now, if I'd have had some, or we'd have had some direction you know, that they'd routinely scanned him early as a neonate, as an infant. The educational plan could have been directed towards that problem. So we're at a, you know, I don't know if there's anything you do at this age, but it seems like a wonderful idea, and I'm sorry it wasn't around when that happened. Also, your supercomputer isn't like me delivering Dan's punch cards, and you'll get them in 4 days. So
Yeah.
there's a lot changed at Ohio State, but I was interested to hear you speak.
That's great. No, that's it.
Of something that would be, you know, that it'll be out there.
Yes, definitely. Yeah. And, you know, and as part of this, right, I think that we always think that earlier intervention is better, and that's certainly the case, right? But that doesn't mean that there aren't things that would be helpful years down the line, right? So we're, as I mentioned, we're also continuing to get cross-sectional data points such that, like at later points in life, such that we can follow up with them as well and look at longitudinal changes when they do get different types of practice and different types of therapies. Yeah, and I think that there's a lot of work to do, but you know, one of the questions that I get when I talk about this stuff is, well, is it practical to scan everyone, right?
Yeah.
And probably not, right? And I'm aware of that. And to that end, what we're trying to develop is a way to understand, you know, how these different brain metrics influence behavior and how they predict behavior. And we're looking at the, you know, relationship with a whole behavioral battery, such that what we can do years down the line is people can, you know, take different behavioral assessments. They can, you know, get them through their school or through the clinic. And the clinicians or educators can upload those scores to our software, our website, and they would be able to get an assessment of, you know, how well we can predict what will happen years down the line as well. And they will get an accuracy, you know, we're X% sure that this may be the case. And as more and more data is uploaded, we will get better and better with our predictions. And so I'm hopeful that, you know, years down the line, we will be able to use, you know, a set of behavioral assessments to inform what the brain will look like without actually having to perform a brain scan.
Interesting how they do.
Are you doing any work with the department or the College of Education, particularly the like the reading labs there? Or I know when I was in school psych, we used to have a child studies center, much different than what you envisioned. But it would seem that if there are people in the college doing similar research, it would be good to work together on those so the teachers could see the implications for their work and vice versa.
Exactly. Yeah. And that's one of my goals with the center is, you know, to first start out with within psychology and to branch out and to include the School of Education as well as medical schools, such that, you know, there's a central place for collaborating and merging ideas. And, you know, we need to be much better at sharing both resources and ideas throughout the college. And I think this the center will be crucial for that endeavor.
Great.
I can ask another question if no one else is going to jump in. What are some of the other, this research questions seem pretty clear cut because of the developmental milestone of reading being something that you could detect differences in the brain area. Are there some other research questions that you see on the horizon that are also interesting? I mean, you had a limited time to present today. What are some of the other areas within this area or outside of it that you're thinking about? as far as research questions?
Yeah. Well, I am looking at different types of school aid skills. So not only reading, but also math, an understanding of numerosity, right, to start, and then the ability to develop math, as well as things like working memory, the ability to filter out irrelevant things in the environment, attentional control, and it's rare to study a whole slew of different processing, but I feel like we have to, right? If we're trying to see, we can't just be stuck in our individual small regions. We have the ability with neuroimaging to look at the brain as a whole. So I think we should be studying a bunch of different mental functions, and a lot of things seem to develop at between the ages of like 4 to 5. So children start to understand that they have a mind that's different from everyone else's, right? So that's the theory of mind. And with that skill, they're then able to make giant leaps in inference, and they're probably better able to learn from other individuals. They're able to be cunning, which definitely changes how they interact and how they as well as their emotional processing. And so we're looking at a bunch of those changes that occur around that age range. And we're looking to see, what makes a child immature in terms of their brain and their behavior, and what makes an adult childlike, right? So what are the types of brain markers that we can pick up on that continue to adulthood? Are they similar processes? Are they different? Right? There are a whole bunch of open questions there with respect to how individuals learn. So yeah, there's a lot of stuff that we're looking at, not just reading. But reading is convenient because of it has a set time point, and it seems like something tangible that we can actually make good headway on and good progress on.
It seems like things are wide open. I mean, lots of things that you can go toward. So you almost have to be a little bit careful not to have too wide of a lens at this point.
Yeah, that's right.
Assuming you and your group aren't the only ones doing this research in the country, that's probably
That's right.
safe assumption. Is there longitudinal data that you've scanned the kid at 1, 2, 3 years old, and you can say, okay, he or she clearly has physiology that is highly predictive of a dyslexic process, and then you find at 7, 8, 9, what have you, that's 80% accurate, 50% accurate?
Mm-hmm.
What's the literature show?
Right. So with brain scans, what me and my colleagues have been finding is about we're able to predict 70% of the variance in reading ability in first grade using a pre-reading brain scan. And my hunch is that we will get much better at making those predictions with more data. And once the pandemic started, what I was doing was reaching out to all everyone that I knew, all colleagues around the US and abroad, such that we could open up our databases with one another and that we could work together. And I've started up multiple collaborations like that. So now what we're able to do is exchange models and see how well we can predict different abilities as these children get older. So yeah, we're getting there.
Okay. 70 is good. Yeah.
Yes, it is, right? Yeah.
How many other colleges and universities are doing this, and which ones, and how many children have you scanned?
Right. So many colleges and universities are doing this. I know the Boston network, for example, has the most. So BU, MIT, Harvard, Tufts, all of them. So far, we have scanned 35 kids, and and that's quite a lot, given that this is my 4th year here, and we spent basically the first year trying to develop everything. Yeah, so it's a hot topic, and trying to predict different milestones and different types of behavior is a hot topic on its own, but reading is also something that is very well studied. So it's interesting because it involves so many different things. It involves basic perceptual visual functions, but it also involves a high-level understanding of semantics as well as working memory. And because it has so many different components, it attracts researchers from a variety of different fields. And so there are a lot of different competing theories, and it's a hotspot for sharing different ideas from different disciplines and different literatures.
Are all the 35 you've scanned all babies? Like, are you going to follow the same cohort, or did you start with some from different ages?
We started with different ages. So we scanned 5 babies so far before COVID started. Right around March, we had set up a very nice collaboration with the Wexner maternity ward. And when everything closed down, we're starting up again, but that sort of put a damper on things. But with the 35 other kids, they're from the ages of 3 to 5, and we've gotten longitudinal assessments on most of them.
Out of curiosity, what sort of metrics do you use, both in the scanning and in the follow-up for this? And did you develop those metrics, or were they established before?
The things that I have talked about today, so the methods were developed before, but the computational methods are the ones that I developed. And the metrics that we're using are the images that we get. So we're using brain function. So how much is a brain region activated or excited about a particular thing that you're looking at or you're hearing. And we're also using strength of connection. So either looking at white matter, physical, pathways that you can sort of follow, as well as functional connectivity, which is sort of an estimate for how things are connected to each other. And that's just, brain regions fluctuate, and it's not at chance. They are fluctuating at a particular rhythm, and those rhythms correlate with one another with different brain areas. So, if every time I clap, you clap too, we're in sync, right? And so that's what happens with these brain areas. So we're looking at metrics of functional connectivity, structural connectivity, as well as functional activation, so Fmri. And we're merging all of those metrics, and we're building computational models to predict either future, what the brain will look like, but also behavior. And those behavioral assessments aren't things that I've developed on purpose, right? So I want to relate what I'm doing to common, neuropsychological or educational assessments, such that this stuff can be actually applied and used. Does that answer?
Yes.
Okay.
And I'm just going to take a moment, and it is 5 p.m. and we were scheduled from 4 to 5. So if you need to leave, please feel free to. Dr. Sagan will stay on if you have any additional questions for a few minutes, but wanted to let you know it's 5. So if you need to leave, feel welcome to, and really appreciate you coming today. And I would be happy to provide Dr. Sagan's contact information if you think of questions after the presentation today. So just let us know.
Thank you for the opportunity. It's been very enjoyable.
Thank you. It's been an honor. Thanks.
Great presentation.
Thanks.
At her age too, the journal. Appreciate
How do you decide you're gonna look at that? Good. Really? Junior,
what
check yourself off, you can sign off.
that's the next stage is okay, what do you do to know when it works, have an idea how it's going to turn
So does
out.
anybody have any additional questions or anything else they'd like to share?
Thank you, Dr. Sagan.
Oh, thank
Thank
you.
you. Good to meet you.
Thank you for being at Ohio State.
Thanks. I'm happy to
Yes,
be
we're
And
happy
the
she's here.
chimes sound like Orton Hall. I'm hearing chimes.
I think your problem is not in what to do. It's just the unlimited focus that you can have on this. We're just talking about what about interventional
right.
activities? You know, you have the ability to project years beyond what forms of intervention might be most effective for X, Y, or Z.
And we had this 27-weeker who was in a incubator under blankets and the room was dark because he was overexcitable. So you wouldn't have gotten him into an MRI screener for a long time. And having been a pediatric nurse for 30 years, I applaud you for getting 5. I mean, that's a big deal. So.
Yeah, neonates are they're actually some of the easier populations just because they do tend to sleep through a bunch of things. But there's between the ages of 1 to 2 are the hardest. And so with that, we just I did some of that at MIT, and I'm going to do it here too. But we just do a lot of time points, right? So we just bring them back in and we have very short scans, about like 30 seconds to 1 minute long. And we just do as many as a child will cooperate through. And
I don't know how your machine is getting them into the machine is so loud, which would be one of the things about the open-ended one. You could just quietly set it there. It's like that startup point. You could reach the baby into the into the chamber. It was like, so that you did that at all is amazing and applaudable.
Thanks. We spent quite a bit of time developing those things.
And getting them to stay still.
Yeah.
I was wondering what your reinforcements for that would be.
For infants, it's,
You just.
Yeah. We
Go
usually
for it.
have an MRI safe pacifier. And so we do get sucking motions from that, and we just do more scans there. But it helps to have the experimenter put their hand on the baby's chest. It gives them a sense of warmth and security, and it also helps us see the child, and see if they're moving, if they're uncomfortable, and so on. But if they are, we try very hard to make sure that it's as quiet as possible. But of course, the scanner bed has vibrations, right, as you're conducting the scan. And so some babies will wake up a little bit from it, and then they will go back to sleep. Some babies just will not. It just depends on the child. And even, across different temperaments, we do make a note of that. And so we are interested in how temperament also affects things. But we give them plenty of opportunities. We give the families plenty of opportunities to come back if they so choose.
The good news is in 5 years, the machine will be half the size, half the money, and will not make loud noise. So just wait for 5 years.
So right now, actually, one of the, I talked about innovative technologies, but one of the things that I'd like to do, and I've been talking to an MRI firm about this, is to develop neonatal head coils that move with the head. And that would, be groundbreaking for the clinic as well, right? Because you don't want to have to sedate children. And so if you were able to do that, it would be easily translatable to the clinic. So hopefully in 5 years, we will be the ones to come out with that neonatal head coil.
Say goodbye.
Have a good evening. Thank you.
Thanks, Kurt. Thanks for being here.
Bye.
Thank you for everything you're doing.
Thanks.
Thank you,
Great
Jane.
questions.
Thanks for being here, Jane.
Thanks, Anna Mary. Thanks again for joining us. Take care.
Thank you.